exams?
Yes, ma'am.
I'm not getting any answers from others. What happened, other students? Okay, anyways, we'll start with the next topic, that is platyhelminthes. I hope all are attending the class, or it's just that they have just logged into it and then they are doing their own work.
No, ma'am.
Only Nilima is answering me. How about others?
No, ma'am.
Okay, good. At least two I can hear. Kirti ready, right? Okay, so we'll start with our class. We will be starting with platyhelminthes in this class. Platy means flat worms. So that's what is the meaning of platy. Platy means flat worms. They are all tapered and leaf-like structures, and sometimes they can be even ribbon-like structures. So that is the meaning of platyhelminthes. If you can see in the picture, you have a tapeworm, you have a liver fluke. The liver fluke resembles like a leaf, whereas a tapeworm resembles like that of a ribbon. So we'll be talking about these flukes and trematodes and cestodes in a more detailed manner. So here, kingdom Animalia, subkingdom Eumetazoa, phylum Platyhelminthes is what we are going to talk about in this class. And this is a classification of the Platyhelminthes. As I've been telling y'all, we'll be talking only about the classes of each phylum. So here, Platyhelminthes has three classes, that is Turbularia, Trematoda, and Cestoda. So what is the examples in Turbularia? We have Dagysia. In Trematoda, we have Fasciola, Schistosoma. And in cestodes, we'll be talking about Taenia solium and Echinococcus. So each one we'll be talking about one or two examples over here. And coming to the first class, that is Turbularia over here. Before going into the proper classification, we'll first understand the general characters of what platyhelminthes are. Why I showed you all the first classification is because we are going to talk about a little bit about Dagysia, Fasciola, and Taenia. So that is the reason why I put this classification picture on the screen. So under Dagysia, we'll be talking about Planarians, and a trematode we'll be talking about Fasciola, and under cestodes we'll be talking about Taenia. So coming to the general characters of platyhelminthes, they are bilaterally symmetrical. That is, I always tell you, bilaterally means you're going to cut them into equal halves. When you cut them into two equal halves, you're getting two halves which is having all the characters. Something like when you're going to cut our human into two halves, you're going to get each parts on either side. So that's called bilateral symmetry. Dorso-ventrally flattened. Dorsal side, ventral side. Dorso-ventrally flattened is the character of this particular platyhelminthes, and they are triploblastic worms. All the three germ layers, that is the ectoderm, mesoderm, and the endoderm layers. So that is why they are called triploblastic worms. Body shape, generally elongated, flattened. They are sometimes ribbon-like or they are sometimes leaf-like. They're very small to moderate in size, varying to, they can either be microscopic, to view them under the microscope, or sometimes you will be shocked to know that they can range up till 10 to 15 meters in length at a stretch. Sometimes people are infected with this trematodes and the cestodes, especially the cestodes. They have a ribbon-like worm coming out through fecus. So that long the cestodes can be. So that is the size 10 to 15 meters in length. Majority are white in color. Sometimes they derive the color by the ingestion of the food. For example, if they are blood flukes, they get the color red because they are ingesting the red color. So they can be either maroon, dark red, or dark maroon in color. And sometimes they can be gray in color or sometimes brilliantly colored. Depends. Anterior end of the body differentiated into so-called head-like structures, something like scolex-like structures. Ventral surface, that is the dorsal surface, ventral surface. So ventral surface consists of the mouth, presence of a great variety of adhesive secretions. So they have to hold on to the body of the host. So if they have to hold on, they have to have a proper rostrum, the suckers. So they have to attach themselves onto the body. That is, they have hook-like structures, or also called suckers. So they have that strong suckers and hooks so that they can hold on to the body of the host. Why? Because they are all parasites. Body covered with cellular syncytial, one-layered, partly ciliated epidermis. What do you mean by this cellular syncytial? That is, all these single cells are closely attached together, forming a syncytial layer-like structures. They resemble a layer-like structures, but multinucleated because the number of cells together, placed together. So they are called syncytial, one-layered, but they are not double-layered, one-layered, ciliated epidermis, ciliated, the presence of cilia on the epidermis. While in parasitic trematodes and cestodes, epidermis is lacking because they are always inside the host, so they don't need all these. Only the Dugitia, that is the Planarians, they have to have those cilia-like epidermis on the outer wall. Whereas the trematodes and cestodes, the body is covered by cuticle. Cuticle, why? Because the cuticle can be sometimes very hard, because they have to evade the digestive juices of the host. So they have to survive even in that environment. So the body is covered by cuticle. Exo and endoskeleton are completely absent because they don't need that, as they are generally soft. Hard parts consist of cuticles, spines, hooks, thorns, or teeth. So they only need all these things. Why? Because they have to absorb the nutrition from the host. The genital pores are well marked in Turbularians, less marked in Trematodes and Cestodes. Acoelomates, that is, true coelom is absent. If you can see in this picture over here, that coelom over here, there is no coelom in between these three layers of ectoderm, mesoderm, and endoderm. Only a digestive cavity is present in the center. Usually, if there is a coelom, then it has to be present in the mesodermal layer, that is in the middle layer of the any individual. Hence, that will be called as true coelomates. But here it is a coelomate, that is, no coelom is present, or coelom is absent. The cavity is absent. That is why it's called as a coelomate. And if you can see the structure of the cestodes over here, you can see a hook on the anterior region. There's a scolex, that is the head part is called a scolex, and the scolex has four suckers you can find over here, because this is a 2D picture. I can't show you the fourth one on the other side. So this is 1, 2, 3, and the other one behind it. Okay. Then it is the scolex is having a neck region, and the neck region is followed by the proglottids. You can see all those division-like structures over here. So that is how cestode resembles. And this is a picture of how one acoelomate resembles. When we are talking about true coelom, I'll be talking a little bit detail about how the coelom is formed. But here you have to remember it is made up of acoelomate, that is, there is no true coelom present. Digestive system totally absent in acilas and tapeworms, but in other flatworms it consists of a mouth, a pharynx, and a blind intestine. Respiratory and circulatory system are absent because they are going to survive in the host, so they don't need much of this respiratory and circulatory systems. Excretory system consists of a single or a paired protonephridia, if you can see over here. So this is a protonephridia, proto early form of the excretory system. The excretory system is called protonephridia. That is, even before the kidneys are formed in the vertebrates, all these animals had this type of nephridial type of excretory system, where they resemble like a flame cells over here, because the cilia is always moving. So it resembles like that of a flame. So that's why it's called a flame cells or protonephridia. And in acela, the protonephridia are absent. Nervous system, primitive, if you can see the nervous system over here, they have a ganglia-like structures, two ganglion-like structures over here, which is a brain. And the brain is continuous with a two lateral nerve cords. And these two lateral nerve cords is connected by transverse nerve cords, so that the whole body is connected in a single structures. So this is a primitive type of the nervous system. These the platyhelminthes has. Okay, so they resemble like that of a ladder type. So ladder type of nervous system is found in this platyhelminthes. And sense organs are common occurrence in turbularians, but these are greatly reduced in parasitic form. Turbularians, that is Dugitia is what we are talking about. And chemo and tango receptors commonly occur in the form of ciliated pits. Chemo and tango. What do you mean by chemo and tango? The receptors for the chemicals that is present in the environment, and tango is the touching receptors where they are moving. What is the temperature of the water or the temperature of the surroundings is a tango receptors. That is, they occur in a ciliated pits and grooves of the platyhelminthes. The sex is united, that is, they are hermaphrodites. You know what a hermaphrodite is? Both male and the female reproductive organs are present in the same individual. Very few are separate. Reproductive system highly evolved or complex in most forms. Reproduction either asexual and sometimes it is sexual reproduction. But asexual reproduction occurs in turbularians. And this is how the structure of platyhelminthes looks like. So this is a digestive system from the mouth. A pharynx opens into the esophagus. From the esophagus, it opens into the intestine to intestine, which is divided over here. And then they have this protonephrida, the yellow color, not the yellow color, that's a yolk duct. The nephrida is not shown over here. This is because of the reproductive system that is seen over here. This is the pink color is the uterus over here. The purple color behind is a uterus. The light purple color is the seminal vesicles over there. And you have a paired testis, vas efferens. You can see a clear picture in the next picture, which I'll show you all. It will be very useful over there. Majority of forms, eggs are devoid of yolk. That is, less yolk, alecithal or less yolk is present in these type of eggs. And they are provided with special yolk cells and they are covered by egg shell, because when the eggs are released into the environment, they have to be protected. So they are covered by egg shell. Cross-fertilization in trematodes and self-fertilization in cestodes are very common. Fertilization is internal, it is not external. That is, the eggs and the sperm is not released outside. It is internal. Fertilization is internal. Life cycle complicated, involves one or more hosts, definitely, because they have to survive. Parthenogenesis and polyembryony. What do you mean by parthenogenesis? That is, the egg doesn't need a sperm to fertilize the eggs. So they are just going to fertilize on their own. That is called parthenogenesis. And polyembryony, that is formation of more than one embryo from a single fertilized ovum. So from a single fertilized ovum, you might get at least 4 or 5 different embryos that is developing. So that's a polyembryony, many embryos from a single fertilized ovum. And they commonly occur in trematodes and tapeworms. Some tapeworms propagate by endogenous or exogenous budding. So budding can be internal or it can be external. That is what they say. Flatworms are either free living or they can be ecto or endoparasites. Okay, so development. Life cycle is complicated in most flatworms with one or more larval stages. Here, the larval stages are most important. You might be asked to write the larval stages of the platyhelminthes. So you will have to give a little bit importance for this larval stages of flukes. So in liver fluke, that is, when you're talking about the trematodes over here, it has different types of larval stages. That is, first one is a miracidium larva. The miracidium changes to sporocyte. The sporocyst changes to rhedia, then to cercaria, then to metacercaria larva. That is a liver fluke. When you're talking about the tapeworm, we'll be talking only about oncosphere, hexacanth, and cysticercus larva are found. Oncosphere, it is spherical in shape. Hexacanth, it is having the the spine-like structures, hexa, 6 spine-like structures, which helps in boring into the host cell, so hexacan, and cysticercus, which is the infective stage of the tapeworm in the vertebrates, wherever which is where it is going to infect. Regeneration, well marked in some flatworms. Example is Planarian larva. Now, if you can see this Planaria worm, not larva, Planarians, that is Dugutia, it's also called as Dugutia or Planaria. Scientists are very much interested in this Planarians because if you're going to cut this Planaria into 2 halves, each half is going to develop into a new individual. So that is the capacity of regeneration this Planarians have. And if not only into 2, if you even cut them into 10 pieces, small 10 pieces, each piece is going to grow its head and a tail. So that is the capacity this Planarians have, a beautiful regenerative capacity. And why the scientists are interested in this Planarians is because if such Planarians can regenerate a complete head and a tail, why are human beings not able to generate their lost organs or limbs? Why are we not able to do that when a Planarian can grow a new head and a new tail? Why can't we do it? So what is that? Stem cells that is triggering them to grow them into a new Planarians. See, each single Planaria, when you're going to cut them into 10, from one Planarian, you're going to get 10 Planarians. How is a real question mark for the scientists, and there is a lot of research going on in that field. And any students in my class who are thinking of doing the research, you can definitely work on this regenerative capacity of the Planarians in your future. After your degree, you can just take up MSc, and then when you're working on PhD, you can do this work. Let's see who's going to get a Nobel Prize for this research if you all are doing. Yes, are you all listening to me?
Yes, ma'am.
Yeah, I want all my students to think on future. Yes, ma'am.
Then how will Planaria will die, ma'am?
Planaria don't die at all. They don't die at all. Because how many times you keep them cutting into small pieces, they will keep growing. And they have very good memory power also. So a new brain also can remember what a old brain had. So you don't have to again feed a new cells into it, new memory into it. It can remember everything. That is, when you're going to cut a Planaria into 5 different parts means a tail has to grow a new head. A body part has to grow a new head. Yes. Those cells will remember what the main head memory has. You don't have to give them a new memory cells. So that is the greatness of the Planarians. Yeah. So as a zoology students, I think you can think on these lines from this stage itself, so that you can complete your MSc, and in your PhD you can really work on this topics over there, so that you people will be given a Nobel Prize if we are going to find out what is that triggering point to trigger the cells, our stem cells, to grow into a new limbs, or whatever the lost limbs we are going to lose. If you're going to get it, then definitely you will be given a Nobel Prize. I hope my students are going to do that in the near future. Yeah. Okay, coming to the classification. Yes, so now you will be understanding what is this classification over here. Turbillaria, that is what we are talking about. Dugutia, that is the Planarian larva, the Planarians. Then, and the Trematodes, will be talking about Fasciola, that is the leaf-like structures, Trematodes. And in cestodes, we'll be talking about the tapeworms, that is the ribbon-like structured worms, all comes under cestode, that is Taenia solium. Okay, so coming to one by one over here, classification. Turbillaria, these are mostly free-living flatworms. Examples are the Planarians over here. These are slow-moving ones in the streams, and they bear cilia on its body. So you can see the cilia present on the outer covering, that is the body surface is covered by cilia, and it has a great power of regeneration. And coming to the trematodes, trema means piercing. That is, they have to pierce the host cell and they have to invade inside. That is why they're called as trematode. Toad means form. Trema means piercing. They can just pierce and get inside the host. So they are ecto or endoparasitic. Examples of here: Fasciola, Schistosoma, Polystomum. Polystomum usually found in the bladder of frogs and the turtles. Fasciola and Schistosoma you can find in all the other vertebrates. Examples of here, they are also called as blood fluke because mostly you find them in the blood cells of the vertebrates. So that's why they are called as blood fluke. The name of the trematodes are named according to where they are present. So for example, if they are present in the lungs, they are called lung flukes. If they are present in the intestine, it's called intestinal fluke. But here it is present in the blood, so it's called as blood fluke. Because they live in the hepatic portal system, that is in the liver. Hepatic portal, hepatic means liver, and mesenteric blood vessels of the human beings, and the intermediate host is snail. Coming to cestodes, cestodes are the tapeworm-like structures. You can see the tape. Cesto means girdle form. Girdle means a belt-like structure. So a girdle is nothing but a belt-like structure. So that is, if you can see over here, so they resemble that belt-like structure over here, or the ribbon-like structures. And they are the endoparasites of the vertebrates. Body is generally divided into few to many proglottids. If you can see those sections of your small sections, so these sections are called proglottids, but they are not true segments like that of annelids. So they are just the divisions over here. And examples of here are Taenia solium, Echinococcus, and Fasciola hepatica. And when you're coming to talk about the example of Fasciola, Fasciola is a liver fluke, that is because they are surviving at the liver, so called liver fluke. Adult Fasciola is found in the liver and bile ducts of sheep, goat, which are its primary hosts. So they are the vertebrates. So the primary host is a sheep and a goat for this Fasciola, and the secondary host is a snail. We have always come across the secondary host is usually an invertebrate. So here the snail is a secondary host for the liver flukes over there, and the primary host is a sheep or a goat, and sometimes it can be even human beings. Coming to important trematode and cestode parasites. So we'll be talking about a little bit about what are the important parasites of the trematodes and the cestodes over here. Trematodes, leaf-like structures. They are flat worms known as flukes. They are obligate internal parasites. Radhika, can you mute? Radhika, please mute your cell phone or whatever you're using. So they are obligate internal parasites. Obligate means compulsory. They have to invade and they have to live inside. So that's why they're called obligate internal parasite with a complex life cycle requiring at least 2 hosts. Intermediate host in which asexual reproduction occurs is a snail, and a definitive host is a vertebrate, definitely because sexual reproduction has to occur in a vertebrate. Infection by trematodes can cause diseases many types of vertebrates, including mammals. We are also not left out. Birds, reptiles, and fish. They are divided into 2 subclasses. What are they? Aspidogastrae and Digyenia. These are the 2 subclasses under this trematode. Aspidogastrae is a smaller subclass with just 61. Platyhelminthes, if I've forgotten, 15,000 species are known to us. All the platyhelminthes put together, we have 15,000 known species. Under this 15,000 known species, Aspidogastrae is a smaller subclass of 61 species. These fluke mainly infect the bivalves and the bony fish. What do you mean by bivalves? The snails, the shells, right? So they are called as bivalves. And of course, the bony fishes, Osteoichthys, all the bony fishes that latest fishes that is surviving in the environment are bony fishes, whatever the fishes that we are eating, because they have bones in them. Then the next one is Digyenia. Majority of the subclass, the species consists of this Digyenia. They need 2 hosts to complete their life cycle. So Digyenia, and 2 hosts here. It's not only the sheep, the animals. It can also include human beings. So Digyenia, they can survive in different types of hosts. It can be an animal or it can be humans. They are majority and they are found in mollusks and vertebrates. Flukes that causes disease in humans are all often classified based on the organ system they infect. That's what I told. To whichever the organ they infect, the name is given to that particular fluke. So for example, over here, blood flukes, because they inhabit the blood in some stage of their lifestyle. And what is that causes this disease? Blood flukes that causes disease in human includes Trichobilharzia regenti is one type of disease that is caused by this type of the blood fluke. And this is also called as swimmer's itch. Swimmer's itch. That is, you will find that when you're constantly in the water, you have those itching sensation between the digits or the toes, the fingers and the toes. You have those itching sensation. It's because of the infection by this flukes, that is Trichobilharzia regenti, which causes the swimmer's itch. You sometimes apply some moisturizers to overcome that itching sensation. Then the next one is schistosoma, which causes schistosomiasis. You must have heard this word. A definitive host are the humans. Humans are infected when the Cercarian larva penetrates the skin. That's why they are called as trematodes, piercing. So they penetrate the skin of the host. Adult blood flukes can live for years in humans or animal reservoir hosts. The next one is schistosoma hematobium or japonicum, which are of particular importance as these are carcinogenic parasites. Carcinogenic means those that causes cancer in the vertebrates which they infect. So usually hematobium infects the urinary bladder and is among the most important causes of bladder cancer, urinary bladder cancer in humans. So you have to be very careful with these type of flukes which infect us, because once they are inside the body, they cause different types. So for example, if they are infecting the blood, the person can become anemic because they are utilizing the blood, and they also causes liver rot or liver cirrhosis because we are losing lots of blood. In turn, they again start degrading the different organs that they infect. So here you have one more called the liver flukes, which are commonly found in the bile ducts, liver, and gallbladder in certain mammals and avian species. And what are the examples of here? They include Fasciola gigantica and Fasciola hepatica. So this is the Fasciola hepatica, the leaf-like structures of here. Ophistorchis felinius, this is one type, and Ophistorchis viverrini is another type. And these Ophistorchis are carcinogenic parasites that are strongly associated with the development of cancer in the bile ducts, that is cancer in the liver. You have in the blood flukes, you have the urinary bladder cancer, while the liver flukes causes bile cancers or the liver cancers, that is the Fasciola hepatica and Ophistorchis. Coming to lung flukes, because they are invading the lungs, so they are called as lung flukes. There are 10 species of lung flukes that infect the humans, causing paragoniamiasis. This is one more important disease, paragoniamiasis, because they are going to affect the person, whomever it is invading. They try to give respiratory disorders, the problems in the respiration process and all. The paragoniamiasis are the ones that leads to liver infections, lung infections. Of this, the most common cause is a human paragoniamiasis, which is caused by Westermanni, the oriental lung fluke. The lung flukes requires 3 different hosts in order to complete their life cycles. The first one is a snail, the second one is a crab or a crayfish, and the definitive host is a human or an animal host. So this needs, the lung flukes need 3 different hosts. One is a snail, one is a crab, and the next is a definitive host, that is an animal, vertebrate animal or a human host. So these are the different flukes. And the last one is the intestinal fluke, because you find them only in the intestine. So intestinal fluke, and they inhabit the epithelium of the small intestine, that is the epithelial region of the small intestine. And they include Fasciolopsis busci, which is a notable parasite of medical importance in humans and veterinarian importance, because these are the one that the symptoms are diarrhea, abdominal pain, if there is a heavy infection of this intestinal flukes. The person is always having diarrhea, that is dysentery and diarrhea, loose motions, what you call them. And of course, the severe abdominal pain by this particular intestinal flukes. So this is the Fasciolopsis busci. And you can see the color is because of the food that is being ingested in the environment in which it is inhabiting. So that was about the trematodes, all the flukes that we talked about, the blood flukes, the liver, the lung, and the intestinal flukes. So if they're going to ask you to talk a little bit about the trematode parasite, you're going to talk about all those different flukes that we have just read. Now coming to cestode parasites. Cestodes are tapeworm or ribbon worms that are present in the form of a ribbon shape. Cestode, a class of parasite worms in the flatworm phylum. Most species are best known are the Eucestode subclass, and they are ribbon-like worms known as tapeworms. Their body consists of many similar units known as proglottids. If you can see the divisions of here, there are divisions of here. So each division, each rectangular shaped small structures, they are called the proglottids. And this proglottid essentially packages the eggs. That is, we have those egg trays where we keep our eggs in one tray. It's something like that. Each box consists of many eggs. They are packaged in many eggs. They are called as gravid proglottids, and they are released outside in the environment, which helps in infecting the other host. Humans are subject to infection by several species of tapeworms if they are eaten undercooked. That is, if you eat a meat which is not properly cooked, we are definitely going to get infected by this particular cestode parasite. Usually, this is affected when a pork means a pig, when you eat an uncooked meat of a pig or a beef, that's a cow or a buffalo, and a fish. So when we are going to eat this uncooked, we can't say uncooked if you're not going to cook them properly. Thankfully, Indian cooking, we give a proper 100 degrees cooking. We put it in a cooker, pressure cook it, again fry it, again heat it. Thankfully, we will be killing all those parasites that are present in the flesh of that particular meat that we are eating. Sometimes when we are eating some not properly cooked food, usually what happens is if you are going to cook it in a microwave oven, or if you are going to do a fast stir fry, we are not going to kill those parasites properly, and when you ingest those, we are infected by this particular tapeworms. So we have to be very careful when you're cooking meat especially. Okay, so what are the different types in the cestodes? Taenia saginata is one type of the tapeworm, which can go up to 4 to 6 meters. My goodness, the size of a sari, that big, 4 to 6 meters long and 12 mm, that is almost 1.2 centimeter broad. It has a pear-shaped head, scolex. I'll show you the picture how they look like. So this is the Taenia saginata. So this is a scolex, pear-shaped scolex, and this is the length. So this is the head region over there, the small one, and this is the length of the Taenia saginata, almost 5 to 6 meters in length, and this is a scolex over here. The Taenia solium also almost same like that, but this is a little bit smaller when compared to Taenia saginata. So what are the features over here? It has a pear-shaped head or the scolex. The head region is also called as scolex with 4 suckers. Have you seen this? So they have these 4 suckers, 1, 2, 3, and 1 behind. The top one is a rostrum, so don't get confused with the suckers over here. So the blue color ones are the suckers. So here you can see 3, but the 4 fourth one is on the behind side, the behind of the head region. So this is the head region, or also called as scolex. This is also here. This is a rostrum, the mouth region, or the rostrum. Okay, so and it has 4 suckers, but no hooks or the neck. It has a long flat body with several hundred segments, are called as proglottids. All those segments, the rectangular shaped segments, are called as proglottids. Taeniasolium, slightly smaller than Saginata, globular scolex with 4 suckers and a circular row of hooks. You can see the hooks over here. So you have those circular row of hooks over here present for attachment. And it has a neck and has a long flat body, which is almost 10 centimeters. So a little bit smaller when compared to Saginata. Here we are going to talk about centimeters. There we talked about meters. Remember that. Symptoms: light infection remains asymptomatic. You don't have any symptoms when you have a light infection, but heavier infection may produce abdominal discomfort. You might have some pain, epigastric pain, vomiting, and of course diarrhea or dysentery. Cysticercosis is what is the infection when you say a person is infected by cysticercosis? Is Taeniasolium eggs can also infect humans and cause cysticercosis, that is resulting in blindness and neurological disorders. Why? Because the cysticercosis, the larva, can invade inside. Once it goes inside our system, it will slowly, through the circulation, it will invade the liver, the lungs, the eyes, and also the brain. And once it infects the brain, then we are going to have neurological disorders. And if it's infecting the eyes, then blindness can occur. So we have to be very careful from this cestodes. This is the picture I showed you all just now. And there is one more called Diphalobotram Latum. This is also one more important disease or the infection. Fish tapeworm infection, which is called as Diphalobotram Latum, distributed worldwide in subarctic and temperate regions, associated with eating of raw or improperly cooked fish. That is, sometimes there you find in the bundis over there outside, I don't know what tawa they keep, what oil they use, and they just put everything, fish, all 4 or 5 fishes together 2 times like that, and then they give it out, and people just hog it. I don't know how they eat those type of fishes. Those fishes are not properly cooked. When it's not properly cooked, we get infected with this longest tapeworm, that is Diphalobotram Latum. The longest tapeworm found was 3 to 10 meters in man, and more than 3,000 proglottids. Imagine that many proglottids, the rectangular shaped structures found, as much as 3,000 proglottids. So that big, 10 meters big, is those longest tapeworm. The scolex resembles 2 almond-shaped leaves. Almond, what is the meaning of almond? The badam shape. Let me show you how they look like. So this is the almond-shaped leaf-like structures of Diphalobotram. That is the head region of this particular tapeworm. And see the size. So this is one big 10 meters tapeworm that you can find. This is another picture which is showing in a close-up. So this is the size. So imagine the size that is present in the humans. And this is the almond-shaped leaf-like head structure of Diphalobotram. And clinical symptoms may be mild depending on number of worms. They include abdominal discomfort, loss of weight, loss of appetite, and sometimes malnutrition. Why malnutrition? Because whatever the person eats, these are being taken away by this particular cestodes. What is left out for us? So the humans will definitely be malnutrition, and they start losing the appetite. Appetite is they start losing the interest in eating. And what happens ultimately? Anemia, loss of blood, neurological problems associated with vitamin B12 deficiency, which is seen heavily in infected individuals. Vitamin B12, very, very important vitamin, because almost all our daily life energies, the daily life interest, the folic acids, everything is present in B12. So I suggest every one of you all to have proper diet wherein they have this particular vitamin B. Vitamin B is very, very important. You can get them in omega fatty acids and also in omega-3 and omega-6 fatty acids. You can almost eat all the nuts, that is your almonds, um, the apricots, the walnuts, mostly the walnuts, the pistas. So try to eat proper food to have all these particular vitamins in your body so that in the future you will not have any problems, right? So this is the picture of Diphalobotram. Now coming to the type study. So we talked about the trematodes and cestodes and also turbularians. Turbularians, not much is given. There is not much of fearful nature in the platyhelminthes turbularians because they are all free living. They are not parasites. So here we'll be talking only about the cestodes and the trematodes. So first coming to the trematodes, the class Trematoda, piercing, remember that always. So under that we'll be talking about Fasciola hepatica. Okay, so the first one is type study is Fasciola hepatica. Classification, kingdom Animalia, phylum Platyhelminthes, that is the flatworms, class Trematodes, stinging type or piercing type of worm, not stinging, sorry, piercing type of the worms. Order Plagiarchidae, family Fasciolidae, genus Fasciola, species Hepatica. Coming to why it is called as Hepatica, because they are found in the liver, so that is why it is called liver fluke. Fasciola hepatica, hepatica liver. Liver flukes are typical digeneian trematodes, commonly called as flatworms or flukes, on account of their flat structures or the leaf-like structures. So if you can see in this picture over here, they resemble a leaf-like structure. So that is why they are flatworms or flukes. Common liver fluke of sheep. So mostly you find them in the sheep. The first trematode was described by Thomas in 1883, long time ago. And they causes fasciolasis, a disease that causes damage to liver tissues and bile duct of sheep. So if the liver tissue is damaged, then what happens? Liver rot occurs, or liver cirrhosis. Cirrhosis is the liver damage, cell damage occurs to the liver. So we have to be very careful from this Fasciola hepatica. Endoparasite, which completes its life cycle to host. Adult flukes are typical parasites of vertebrates, and the other life history, it has to definitely spend its life in a molluscan or a snail. They have a digenetic life history, so that's why it's called digenes. Okay, for which the group they belong to is known as digenia. Di means two. Sometimes adult flukes invades other vertebrates like not only sheep, they can also infect the goat, the horse, dog, ox, man, monkey, everyone, any vertebrate it wants to. And the seriousness that it causes, it can be very serious, life-threatening, and they cause liver rot because they infect the liver. So liver rot occurs. A single sheep may harbor as many as 200 liver flukes. If you can see in this picture over here, this is the picture of a liver and those patch-like structures over here. So these are the flukes, the digeneian flukes over here. So here you can find only 5 of them. But what he says, as many as 200 liver flukes, it can harbor. One single sheep can harbor 200 liver flukes, and they are cosmopolitan in distribution. Where sheep and cattle are being reared. That is where they grow the sheep and the cattle. If one is infected, all the sheep or all the cattle can be infected. If you want to really see this Fasciola hepatica, you can just go to any slaughterhouse where they have this slaughters, sheep slaughter or goat slaughters. There you can collect some liver where you find the infections over there. You can definitely come across. As a zoology student, you have to be very much interested. When we say you can see something like this, it is your duty to go find out some sheep or a goat that is infected, take out its liver, and you can find those fasciola hepatica clearly seen in the on the liver. So I hope at least one of you all can just go to the mandi, find out any infectious liver. If you find this liver flukes, then definitely take a picture and put it onto your WhatsApp group so that others also can see how they really look like. Because seeing in this picture, you will think that, okay, everything is bought from the Google. But if you see them live, your interest grows even more. So, because here in zoology, it's not just, we are not just talking about diseases and infection, but we are also talking about how they look like, what, because they are also some individuals. So you have to know how, what is their life cycle, how they survive, and what are the organs they have, and how they are able to survive. Endo, that is inside a vertebrate, how are they able to survive, and what are the organs that they are having for them to survive. So these are some important points we'll have to remember. Because sometimes if you want to know the true nature, you have to think yourself that you are in its form and understand how they are able to survive. Okay, so there are some simple interest, self-interest. Okay, external morphology. They are soft-bodied, flattened leaf-like animals. So you can see a flattened leaf-like structures, bilateral symmetry. So here you can see, if I'm going to cut this fasciola into two equal halves, I'm going to get two equal parts on either side. So that's why it's bilateral symmetry. They measure up to 1 to 2.5 centimeters, very small. Width is about just 1 centimeter. The anterior end of the body is drawn into a conical projections called the head region over here. The head bears a small triangular aperture called the mouth. The mouth opens into a small pharynx, and that opens into a esophagus. The esophagus again goes beyond a little bit, and they divide into two branched intestinal limbs. So the yellow color ones is the branched intestinal limbs over there. The intestine again branches into small CK. So these are the small branches called the CK that is present. On the ventral side, you have an acetabulum, one more oral sucker that is called as an acetabulum. This is a little bit bigger than the mouth on top over there. So this is the oral sucker, which helps in sucking the digestive juices from the host. Okay, and between these two suckers, the mouth and the acetabulum, you have a gonophore over here, the genital opening that is called a gonophore. And at the extreme end, there is an excretory pore, and this excretory pore is not connected with the intestine. So they are connected with a protonephridium over there. So intestine is not having a direct opening to the exterior excretory pore. Okay. Internal morphology, the outermost region of the epidermis forms the spines. You can see the spines, small spines that is arising, and it has a basement membrane. Circular, longitudinal, and oblique type of muscle fibers that are present. They have a circular, longitudinal, and oblique muscle fibers, and a number of unicellular gland cells with protoplasmic processes. Gland cell for secretions. They need to secrete adhesive secretions. So they have those unicellular gland cells with protoplasmic processes. That is the internal morphology. Coming to the digestive system, the mouth or the oral suckers leads into the small round pharynx. I've already told all this. It has a muscular suctorial process called the acetabulum. The esophagus is followed by the intestine, a branched intestine. And the intestine is divided into two main limbs, the right and the left limbs. And they are branched into blind branches called the CK. And intestines and its branches are very conspicuous as they remain filled with decomposed products of the blood on which the fluke feeds. That is, they eat. Why? Because they are surviving on the liver. Liver means maximum amount of blood is present in the liver. So they have to eat the, they take up the blood as their nutrition. So the decomposed part of the blood is present mostly in this intestine, where they give that specific red color to that flukes. And anus is absent. There is no aperture of communication between the intestine and the exterior. The exterior is done by protonephridium. So we'll be talking about the excretory system over here. So this is the excretory system. All those red color spots are the flame cells, the protonephridia, we call them flame cells. Why flame cells? Because the nephridia have those cilia-like structure which keeps moving. So that's why I told you all it's flame cells. All these flame cells are connected by this small set of connections over here, which ultimately opens into a main longitudinal excretory canal over here. So excretory system is a branching system of vessel, that is the water vessels that ramify mouth throughout the body. So it's completely throughout the body, they are present over there. From the anterior region of the main canal, 4 sub-canals, so 4 branches are given off, and that 4 branches again branches repeatedly to form small capillary branches over there, and these are connected by that small tiny pink color flame cells, which is also called as protonephridia. Early type of excretory system is called protonephridia. The flame cells excrete the water and waste metabolites such as the unwanted iron from the hemoglobin. The RBCs contains hemoglobin. Yes, somebody has a doubt? No? Okay. The iron, unwanted iron, because they need only the proteins of the hemoglobin and not the iron part. Your RBCs contain also iron. That is why it is red in color. So they don't need that iron. They only need the protein part of the RBCs. A curious factor in the fasciola is that its excretory canals normally contains an abundance of fat droplets. Now, can you imagine why we have so many fat droplets? Because these are ingesting a lot of hemoglobin, and it is present in the liver, and the liver has a little bit of fat present in them because they have those juices to digest the fat. So probably, so that is the reason why this excretory canal normally contains an abundance of fat droplets. So this is how the excretory system looks like. So these are the small dots are the protonephridia. This is the main canal, excretory canal, which opens into an excretory pore. And these are the small flame cells, the small dots, which are connected to the excretory canal over here. That is with the tubes, the terminal capillary tubes opens into the excretory ductule. And this is the flame cell. If you can see in this picture, this is the cilia. And this cilia is the one that keeps moving, resembling that of a flame. The outer side is covered by a pseudopodia, the basal granules, and ultimately they open into the terminal capillary, which opens into the excretory ductule. So they open up into this small tubule, excretory ductules, and this ductule opens into the main excretory canal. And from this canal, it is thrown out from the excretory pore. So that is the reason why the intestine is not attached to excretory to the exterior directly, but it is connected by this protonephridia from the excretory canal. As multicellular systems evolved to have organ systems that divided the metabolic needs of the body, individual organs evolved to perform excretory function. So each one has different organs for performing different actions. So that's what whatever I told just now. The cells in the tubules are called flame cells, which are also called as protonephridia because they are having cluster of cilia. So this is a cluster of cilia which flickers like that of a flame, and the cilia propels water matter down to the tubules. So the cilia propels water matter, whatever the water is present, it propels into the terminal capillary, into the excretory ductule, and through that it is going out through the excretory pore. And during this suction of all this water matter, cilia also draws water from the intestinal fluid, allowing for filtration. So that is, whatever the valuable metabolites are there, they are again reabsorbed, and the rest of the thing is thrown outside, and thereby they maintain the osmotic balance. What do you mean by osmotic balance? Osmoregulation. They have to maintain the salts in the body along with the water content. So both of them have to be in a proper balanced position. Otherwise, what happens is the osmoregulation goes for a toss, as when we have this excess water loss or excess water retention happens if the excretory system is not working properly. Coming to the nervous system, so you have a cerebral ganglion over here on the head region. You can find a cerebral ganglia over there, which is connected by a central ring. This central ring again gives rise to different branches over here. You have two lateral nerve cords. So this is the branches of the nerve cords. And you have another two lateral dorsal nerve cord and a ventral nerve cord. So two dorsal running on top like that, and two ventral running below, and two lateral going on either side. So this side lateral, and two dorsal over here, and two ventral like that. So this is how they get, they are being connected from head to the posterior part. They are being connected, and each nerve cord gives rise to peripheral branches of the nerve cords like that. So they are branching over there, so that all the body parts is connected. Kirti Reddy, you have any question, ma? Because you're please mute it, ma. Okay. Absence of sense organs is due to parasitic mode of life, because they don't really need proper sense organs, because they are going to just survive in the host body, and everything is ready made. The food is ready made. They just have to suck the food, eat it, and survive happily in the host. So not good enough sense organs. They don't have to have those tactile antennae, the sense organs, the chemo receptors. They don't need all those things. Only thing is, if they have an acetabulum, the suckers, and if they have the reproductive structure enough, they eat and they survive. Reproductive system in Fasciola hepatica. If you can see in Fasciola hepatica, I'll show you a bigger picture over here. So this is the reproductive system of the male over here. So here you have those testes, the testes opening into sperm duct. The blue color ones are the male reproductive organs over here in Fasciola. So this is the sperm duct, that is a blue color ones. The testes connected to the sperm duct, and you have two sperm duct on either side. Those directly open into seminal vesicle, and the seminal vesicle opens into the cirrus. So this is the cirrus, and then they open into the male genital pore. So that's the sperm duct, the vas efferents and the vas deferens, the seminal vesicle, and the cirrus of the male reproductive organ. In the female reproductive organ, it has a single ovary over here, so they don't have branched one. A single ovary over here, and thus ovary opens into an oviduct. The pink color ones is the female reproductive organ. The ovary opens into an oviduct, and this is again opened by a Laurel's canal over there. And this oviduct opens into a median vitelline duct, which again opens into a yolk reservoir, where the yolk is being produced for the developing eggs. And these are again connected by transverse vitelline duct. Vitelline duct, why? Because they have to get the yolk production is done by vitelline duct. So it is connected, the yolk reservoir is connected by a vitelline duct, transverse vitelline duct. And you also have a Melis gland, the pink color around the oviduct is a Melis gland. Now this gland helps in production of egg shell of developing eggs. So the Melis gland produces the egg shell, whereas the vitelline duct produces the production of the yolk, secretion of the yolk is done by vitelline duct. And here it is a yolk reservoir. Why yolk reservoir? So that the developing egg is covered by this yolk. Now, what is the importance of a yolk? Because they need the developing zygote or fertilized egg needs food for its survival. So definitely a yolk has to be present. Now this opens into the uterus, and the uterus is having lots of egg. You can see those dark white and black spots over there. So these are the eggs in the uterus, and the uterus opens into the female genital pore outside. So this is the reproductive part of the male and the female of fasciola. Coming to the life cycle. Now coming to the life cycle, here you're going to talk a little bit about this larval stages of fasciola, and we said that it is having 4 or 5 larval stages. Quickly, I'll just show you how it is happening over here. Once the eggs are thrown out from the feces of the humans or the cattle, through the feces, the eggs are thrown out into the environment. This is an unembryonated egg which are passed into the feces. Once outside, they become embryonated. It becomes a hard shell over there. Why? Because they have to survive the hard environment outside until a intermediate host takes them, and further asexual reproduction occurs over here. Now, once the egg is formed, that means sexual reproduction has completed in the primary host, and the egg is formed. The egg has been released outside. Once the egg is released outside, it becomes embryonated. And once they are embryonated, a metacercarian larva is developing inside, and they hatch out. Once they hatch out, when they are swimming in the moist area, and when a snail comes in contact with this miracidium larva, the miracidium larva bores itself into the flesh of a snail. The flesh of a snail is very soft, and the miracidium larva has an apical sharp structures on its head region, and they bore itself into the snail muscles inside. And once the miracidium enters inside the snail, the miracidium changes to sporocyst. The sporocyst gives rise to redia larva. The redia larva again gives rise to cercaria larva. Once the cercaria larva has been produced, they are free swimming larva. They come out of the snail, and they are free swimming larva over here, cercaria, and they come in contact with any vegetation. They get attached, forming a spore-like structure. And if this grass is eaten, or a green leaf vegetable is eaten by any cattle or even a man, if you're not going to wash the green leaves properly, you might have some spores present on them, and this enters into the primary host, and again the life cycle starts once again. So this is the life cycle. We'll go a little bit in detail about what is the larval stages. So fertilization, fasciola is hermaphrodite. As I said, both the male and the female reproductive organs are present in the same individuals, hence hermaphrodite. Cross fertilization is the general rule. There is no self fertilization. It has to undergo cross fertilization, even though they are hermaphrodites. And the copulation takes place in the bile duct of the host, that is in the liver. All this is fertilization is occurring over here. The sexual reproduction is taking place in the liver. Release of fertilized eggs from the primary host, that is after being released from the ovary, the eggs are fertilized either in the oviduct or within the ootype. I've shown you all. So this is the ootype or an oviduct where the fertilization occurs. One pole of the egg shell bears a small lid or operculum for the exit of the future larva. So you have an egg, they have a cap-like structures. So it has to open up so that the larva is released outside. So one pole of the egg shell bears a small lid or operculum. Operculum is lid-like structure which opens, right, for the exit of the future larva. Eventually, the egg leaves the flukes body through its gonophore. So you have a gonophore over here. So from the gonophore, the eggs are released outside and passes down the bile duct of the sheep into the intestine. From the liver, they go into the intestine. From the intestine, it is discharged out through the anus as a from the feces. The egg can survive only if it falls on damp soil. So if they're going to fall on the damp soil, they're going to survive. Otherwise, they're going to get degraded. So what happens? Once onto the damp soil, a miracidium larva is produced from the spores. The miracidium larva is produced. If you can see a miracidium larva over here, it has an apical papillae. So this is an apical papillae where you have those bore-like structures which can bore inside the snail, and they are somewhat conical shape, covered all over by vibratile cilia, vibratile moving cilia, that is vibratile cilia. A distinct head lobe or apical papillae situated on the broad end, and behind the head lobe there are two spots or spots of pigments, which is also called as eye spots. So these are the two spots over here. And of course, they also have this excretory pore, sac-like structures over here, the excretory pore. This is the excretory sac-like structures, which is having that flame cells over here. And the germ balls are present over here. The germ balls from where the next larval forms will develop from here, from this germ balls. The next larval stages will be occurring. So imagine over here, from one single cell, you can see that there are many polyembryony. If you remember, we were talking about parthenogenesis and polyembryony. So this is what is the example over here for polyembryony. That is, from one egg, they are going to produce too many embryos. So how many embryos they are going to give rise to, we'll be talking about. And this is the epidermal plates, the muscle layers, the subepithelium over here. So this is the most important. Apical papillae, the cilia surrounding, because they have to swim. And they have two eye spots, the spots of the pigments of the eye spots. They also have the excretory pore, a small, the brain present over there, the flame cells, and a sac-like germ balls. Okay, so there, it has one pair of flame cells. So you can see a pair of flame cells on either side, and sac-like intestine. The head lobe bears penetration glands, and the miracidium larva swims freely in the water or crawls over the damp surface until it reaches the snail. So once it reaches the snail, the apical layer, the apical head region is going to bore inside the snail, and they're going to get inside the skin of the snail. Now you can see over here at this part, the apical papilla of the miracidium is boring itself into the snail tissue. See the way it is going to bore itself. They're going to make a cut, and very slowly they are going to move inside the snail tissue region over here. So once they have moved inside, the miracidium larva changes. This miracidium larva changes into a sporocyst. The second larval form is a sporocyst. So they give rise to the sporocyst. The miracidium larva, on encountering a snail, bores inside, and they get into the pulmonary sac of the snail. That is the respiratory region of the snail. They're going to get inside over there. There it is going to metamorphosis into sporocyst by casting off its ciliated covering. So the ciliated covering, what is the ciliated covering? You can see the cilia over here. So this is called a ciliated covering. So it's going to shed the ciliated covering, and they're going to form a sporocyst. Now the sporocyst is elongated with an internal cavity containing germ balls. So you can see the small, so this is the internal cavity. It has a germ ball over here and a pair of nephridia, or the flame cells. And these are from the germ balls, the the next larval form, that is the redia larva, is produced over there. So these are the two redia larva that has already been produced, and these are the germ cells which will give rise to more future redia larva over there. So the germ balls eventually undergo a process of cleavage, and from the cleavage resulting in the formation of redia larva. So here you should remember that an asexual type of reproduction is going on inside this sporocyst and also the redia larva. So here in the sporocyst, because of the germ balls they undergo the cleavage, and they give rise to many number of redia larvae over here. There is no budding. It is only the germ balls being cleaved, and there is no sexual reproduction occurring over here, but they're giving rise to number of larval stages. So that is one more example of polyembryony over here. 5 to 8 redia are usually formed in each sporocyst. So one sporocyst is going to give rise to 5 to 8 redia, the small structures inside. So this is the redia larva over here. So each sporocyst is going to give rise to 5 to 8 redia. And this redia, what is happening? The next stage over here, elongated 1.3 mm to 1.6 mm in length, and ventral process. They have two processes. One is ventral processes. So this is the ventral processes called lappets or procruscula. So this is called procruscula. That is the ventral process near the posterior region, and a birth pore near the anterior region. So here the formation of the germ cell occurs, and in the birth pore, from the birth pore, the next larva, that is the Cercaria larva, moves out. So that is the importance of this processes, the ventral processes and the birth pore near the anterior end. Redia has an anterior mouth. So it has an anterior mouth, a pharynx in which numerous pharyngeal glands open. A sac-like intestine is present, and there's a pair of protonephridia. You can see that small pair of protonephridia, one over here and one over there, with two pairs of flame cells. Its cavity has germ balls and the germ cells. So all those round ones are the germ balls or the germ cells, which gives rise to the next larval stages called the Cercaria larva. The redia has the germ cells of the redia gives rise to Cercaria larva. Each redia forms about 14 to 20 Cercaria. My goodness. So that's why it's called polyembryony. Now, can you see that? This sporocyst gives rise to 5 to 8 redia. And this redia is going to give rise to how many Cercaria larva? 14 to 20 Cercaria larva. That's why it's called polyembryony. So the next stage over here, once the Cercaria larva is well developed, what happens is it comes out through this birth pore. So this is the Cercaria larva, two Cercaria larva you can see, and this Cercaria larva comes out through the birth pore of the redia. So it's oval in structure, about 0.25 to 0.35 mm long. It has a long tail and oval body like structures. It has all the rudiments of organs of the adult. There are two suckers, one the mouth over here and the acetabulum over here, and it has a proper alimentary canal consisting of the mouth, the buccal cavity, and the gut cecum, and wherein the esophagus is bifurcating into two intestine. There's an excretory bladder with a pair of protonephridia. So it has this is a pair of protonephridia canals with a number of flame cells. So it has number of flame cells. Earlier we saw only few flame cells. Here it is going to have number of flame cells. Now this is a free swimming larva because it has come out from the birth pore of the redia. They move actively with the help of the tail. So because it is having a tail, proper tail over here, it is going to come outside. They're going to swim freely, and they move out of the snail's body. Then losing their tail, they become encysted. Once the tail is lost, they become encysted, and the Cercaria in an encysted condition is called metacercaria larva. So this metacercaria larva, the time taken in the snail from the entry of the meracidium, the first larval stage, till the Cercarian larva takes up to 5 to 6 weeks. That is almost 1.5 months from the moment the metacercaria infects the the snail, and by the time the Cercaria comes out, it takes about 1.5 months for the development to occur. And you can see all the different parts of here in the metacercaria: the mouth, the acetabulum, the oral suckers, the pharynx, the intestine, bifurcated intestine, the flame cells on either side, or the protonephridia for the excretion purpose, and of course the excretory vesicle or the bladder that is present. So what happens now once the metacercaria is formed? That is from the cercaria, when the metacercaria is formed, again it is going to infect a new host. That is, the metacercaria is going to get attached, because cercaria is a free swimming larva which has come outside, and they form into a cyst like metacercaria. This metacercaria is attaching itself onto any freshwater grass. Wherever there are grass, they get attached to them. And once the sheep eats this particular grass, the metacercaria that has been attached infects the sheep or infects different vertebrates, human also possibilities. So that's how the life cycle occurs in this particular fasciola hepatica. And what is another important one I want you all to remember is fasciola's development stages undergoes 2 rounds of asexual division, where all it is occurring, if you remember, once in the sporocyst for the formation of the rhedia, and once in the rhedia for the formation of cercaria. Okay, so 2 asexual division occurs, and this greatly increases their number, and thus they have a good chance to complete their life cycle, because not all cercarian the miracidium are going to survive the outside world, and not all metacercarians are going to survive in the host, in the environment. Some will get damaged, some will die, some will degrade, but only few of them enter. Even if one enters, it's enough for them to produce n number of parasites inside, because one fertilized egg is going to give rise to how many? 30 cercarian larvae. So thus one egg has a capability to produce dozens of sexual adult in the definitive host, and on the other hand, a single fluke may produce 50,000 eggs. Just one single fluke is going to give rise to 50,000 eggs. So that is the life cycle of the fasciola hepatica. So shall I continue, or shall we do this tomorrow? Because this is going to take another half an hour. Yes, ma'am. You want me to continue tomorrow? Or shall I finish it?
No, ma'am, you can continue.
Continue? Okay, just give me 20 minutes, I'll finish up, because we have only Taenia solium to complete now. Okay, coming to Taenia solium, class Cestoda. Till now, what we talked about? Flukes, they were of class Trematodes. Now we are going to talk about class Cestoda, Cyclophilidae, Taenidae, that is family is Taenidae, genus Taenia, species solium, Taenia solium. Now you can see it is having a scolex, head-like structure called the scolex, and the scolex is having 4 suckers over here. So these are the 4 suckers, and that the center one is a rostrum, or the mouth opening. So as and when I'm talking about the scolex, remember that is the head region, and the scolex is always having 4 suckers and one the rostrum for attachment purpose. Okay, suckers are for sucking the digestive juices, whereas rostrum is helping them to attach itself onto the host flesh. Okay. Found throughout the world, commonly seen in countries where pig is eaten, pork is eaten. Definite host humans, and intermediate host is the pigs, definitely, because that is the reason why when you eat a pork, you are infected with this tapeworm. Humans acquire the cyst through consumption of uncooked food, and it lives in adult stage in the intestine of man. So you find them in the intestine of man. Causes injuries to the mucous membrane lining of the alimentary canal. Why? Because it is adhering by the help of the scolex, that is the head region, and they cause abdominal pain, weakness, loss of weight, and loss of appetite. You don't feel like eating anything much. However, disease caused by this worm is called taeniasis. So that's right, Taenia solium, taeniasis. Body is long. You can see how long this body is. Dorsoventrally flattened. That's why it's a ribbon-like structure, and reaching a length of almost 2 to 3 meters. Color of the body is white or opaque. It consists of a scolex, head called the scolex, and followed by the neck, and the neck is followed by divisions called the proglottids. And these proglottids are of 3 types: immature proglottid, mature proglottid, and gravid proglottid. What are the meaning of all this? I'll be talking about that. Now coming to scolex, if you see, this is the head region, the rostrum on top. So This is the rostrum. Sorry, this is the rostrum. The head region from top. You're viewing this from top. This is from the side region, and this one is from the top region that you're seeing. So this is a rostrum. A rostrum is having 2 rows of teeth or the small hook-like structures. The inner circle having larger hooks and the outer circle having smaller hooks. If you can see this, the inner circle having larger hooks and outer one having smaller hooks. And the head region have 4 suckers. So these are the 4 suckers, 1, 2, 3, and 4 suckers over there. And with the help of these suckers, they are able to suck the digestive juices from the intestine of the host. And these are the hooks, the shape of the hooks. Why they are in that shape is because they are able to hold on to the host intestine. The neck, it is a small neck, thin, narrow neck, which is not segmented. Only after some distance, you can find the segmentation occurring. So these are the immature proglottid, which is immediately after the neck. They are all the immature proglottid. Coming to the strobila. What is a strobila? That is, strobila is nothing but proglottid, or strobilization is from the neck region, new proglottids are being formed. That's called strobilization, or production of new proglottids is called strobilization. The strobila of a mature tapeworm measures almost about 800 to 900 proglottids. Now, if you're going to count these proglottids, each tinea solium can have 800 to 900 proglottids. A linear arrangement of repetitive units. Lydia, ready? Mute ma. The proglottids internally remain connected together by muscles, excretory vessels, and the nerve cords. That is, all these will be shed out in the near future. That is, the gravid proglottid will be cut off as and when the eggs are being produced. But they are loosely connected with each other by muscles, excretory vessels, and the nerve cords. So what is the difference between an immature, mature, and a gravid proglottid? Immature is the proglottid that is present immediately behind the neck region is a immature one. No reproductive organs are present. So that's why it's called the immature proglottid. Almost 200 anterior proglottids are present. So if you can see in this picture, so 200 immature proglottids are present. After that immature, you find the mature proglottid. And after the mature proglottid, you have a larger gravid proglottids. So what is a mature proglottid? Nearly 100 to 200, 150 proglottids. That is, they are going to have only male reproductive organs. No female reproductive organs are present in the first stage. That is, the first development of the reproductive organs is a male. After that, the female reproductive organs develops. That is, 100 to 150 proglottids are mature ones. But then in the later stages, when you're going to count around 300 to 400, slowly the female reproductive organs are also produced over there. And once the female reproductive organs are produced, fertilization occurs. Once the fertilization occurs, the proglottids become gravid. Gravid means excess of eggs are present in that particular proglottid, and that is why it is called gravid. Gravid means heavily infested with eggs. So that's called gravid. Almost 150, 200 proglottids are present in a gravid. That is packaging of eggs, what we called. So that is called strobilization. That is proglottids, gravid proglottids. Now, if you can see in this picture, mature proglottid. In a mature proglottid, you have more, both male and the female reproductive organs over here. So this is the testis, this is the ovary, and the uterus over there, the long uterus over there, the ovary over there. So both male and the female reproductive organs are present in the mature proglottid. Whereas in a gravid proglottid, this uterus, this long uterus becomes completely infested with eggs, and they branch like this. So in this gravid proglottid, you will not find any reproductive organs, but only the uterus that is completely filled with eggs and highly branched over there. So this is a gravid proglottid, and this gravid proglottid is the one that is being pinched off from the tinea solium, and then they are thrown out of the body. So this is what they look like. So this is having the reproductive organs, male and the female reproductive organs, and this is a gravid proglotid where only the eggs are present. And as and when the eggs are full, they are pinched off by a process called apolysis. Apolysis is 4 to 5 gravid proglotid is pinched off, and they are thrown out from the body of the host, and they are thrown out in the form of feces, so that the eggs can go outside and they again form a search for a new host, and the life cycle begins. Nutrition, alimentary canal is altogether absent. Why? Because they are parasite, and the nutrition is saprozoic. We talked about holozoic type of nutrition, wherein the whole food is engulfed by the protozoans like that, or even the poriferans. The whole individual was engulfed like that. But here in saprozoic type of nutrition, the food is sucked. The sucking digestive juices that is sucked is called saprozoic type of nutrition, or the liquid form of food that is taken up is called as saprozoic nutrition. Okay. Digested nutrients diffuse directly to general body surface. Respiration, anaerobic type of respiration. Why? Because in the intestine, you don't find much of oxygen present. Only through the blood, the oxygen has to come. But the intestinal walls, Lydia Reddy, can you please mute the... Somebody call her up and ask her not to. Please ask her to mute the... Okay, respiration is anaerobic because there is no proper oxygen in the intestine. But as and when they get oxygen, the tapeworms try to take in the oxygen and develop faster. That is, the production of the proglottids is done whenever there is lots of oxygen present in the intestinal region. Nervous system, you can see over here, there are 2 cerebral ganglia in the tinea solium. So this is one cerebral ganglia, this is another cerebral ganglia. So they have 2 cerebral ganglia. Ganglia is nothing but a tuft of neurons present in a bulb-like structure. So that is what you call them as ganglia. Now this cerebral ganglia is attached by a transverse connective. So this is a transverse connective. And this transverse connective is again branched into main lateral nerve cords. And in the anterior, you have this anterior nerve cords. And on the dorsal side, as I told you, this is dorsal, this is ventral. The dorsal nerve cord, and it also has a ventral nerve cord. Then you have the lateral nerve cords. And all these are connected with each other. And the whole body is connected together by this nerve cord, so they don't need special sense organs, but free nerve endings are there, so they are connected with that nervous system over there. Excretory system over here, if you can see, they have a nephridial place over here. You have 4 longitudinal excretory vessels. That was the 4 longitudinal excretory vessels are the dorsal vessel and the ventral vessel. The ventral vessel is continuous on the proglottids over there, the gravid proglottid over here, it is continuous, whereas the dorsal one remains only on the anterior part of the tinea solium. And the 4 excretory vessels are joined in the scolex by the nephridial plexus. So this is the nephridial plexus. All the 4 vessels are connected at the nephridial plexus towards the scolex region. And the ventral excretory vessels are unequal in diameter, and they're joined by the transverse excretory canals over here. So each proglottid is connected in this way. And the long cilia of flame cells flicker and maintain the hydrostatic pressure by which water is driven into the excretory canal. So that's the excretory system of tinea solium. Reproductive system over here, same over here. So these are the small dots are the testes. All these testes are connected by vas efferents, that is a small capillary-like structures. Each test is connected by vas efferents. The vas efferents is connected by vas deferens. All the small capillaries are connected by a larger capillary over there. So that's all vas deferens. And there is a seminal, cirrus over there, which opens outside through a common gonophore. And in the females, you have the a pair of ovary on either side, which is connected by an oviduct, and the oviduct opens into an ootype over there. The ootype is surrounded by Mallis gland. Now you know what a Mallis gland. What is the function of a Mallis gland? Is to produce the eggshell, and this opens into a vitelline gland through vitelline duct for production of the yolk, and they open into the uterus over there. So and in the uterus they have this fertilized egg capsules. So the eggs are being present over there. So that is the male and the female reproductive organs of the Taenia solium. And the uterus, yes, so this is a uterus. Once the uterus is filled with eggs, this is how a gravid proglotid looks like. So it only has a genital atrium. The uterus is completely large, branched, containing so many eggs. So this one slot is called a gravid proglotid. And this type, you have 4 or 5 gravid proglotid, which pinches off through apolysis, and they are thrown out into the environment. Life cycle, it's a hermaphrodite, produces by self-fertilization or cross-fertilization. But in trematode, it's only cross-fertilization and no self-fertilization. But here, a self-fertilization and a cross-fertilization can occur. And once the zygote is formed by the fusion of spermatozoa and the ova, the zygote undergoes holoblastic and unequal cleavage. What do you mean by holoblastic? Holoblastic is cleavage from the vegetal pole of the egg. So you have an egg, wherever the zygote is present is called the animal pole. The opposite is the vegetal pole. The vegetal pole first gets cleaved, and the cleavage is unequal. A perpendicular cleavage occurs. So you have unequal sizes of the cleavages. So you have a megamer, mesomer, and a micromeres. That is, mega is a larger cleavage, medium one is a mesomer, and a smaller one is a micromere type of cleavage that are present. And each cell of this can develop into different types. What are they? The megamers develop into a syncytial layer. I told you all syncytial layer is number of cells attached together, forming as though they are multinucleated, but they are not multi. Each cell is attached together. So they form the syncytial outer embryonic membrane. Mesomeres forms radially striated inner embryonic membrane or embryophore, and the micromere becomes the morula stage. And this morula forms into a 6-hooked embryo known as oncosphere or hexacanth. Remember the larval stages: oncosphere, hexacanth. The 6-hooked stage larva. Hexa is 6-1. So 6-hooked larval stage is called a hexacanth, oncosphere or hexacanth larva. And a gravid proglotid can contain 50,000 embryonated eggs. So you can see how many eggs are present over here in this mature gravid proglotid. So each proglotid can contain 50,000 embryonated eggs. So this is how you can understand the life cycle over here. What happens once the proglotid releases, the gravid proglotid releases the eggs in the feces. The eggs move out and they get embryonated. And once embryonated eggs are ingested, what happens is they form into an hexacanth, that is 6-hooked embryo. Now this 6-hooked embryo, once it is ingested into the cattle, what happens is they enter inside and the outer covering is shed out, and it forms into a cysticercus in the muscle tissue. Now this is when we eat the uncooked meat, this cysticercus invades into a human body, and from there they form an adult Taenia solium. So this is how a simple picture I have shown you all over here. And what all I've spoken about is what this picture is all about. So you can just go through this matter that I've given over here. So what happens over here? What is happening over here? Once ingested, the cysticercus, the outer covering is ingested or it is being removed. And once it is removed, the invagination that is found, this resembles like that of a bladder worm because they resemble like a bladder. Once inside, uh, the uh, the bladder swollen up. Once it is swollen up, the invagination becomes exvaginated. That is, it is like that. Now, as the bladder is enlarged by the digestive juices, the invagination becomes exvaginated. They come out. And once the exvagination occurs, you can see over here, the scolex is formed with a neck and a bladder. And slowly, all the different organs are formed over here. And the scolex, the neck region, starts to divide into strobulation, and this strobula becomes into an immature, mature, and a gravid proglottid. So this is called strobilization. So this is how they are going to infect. The surviving oncosphere preferentially migrates to the striated muscles as well as the brain, liver, and other tissue, whichever they love to, and they form the cysticerci. Humans are also accidentally secondary host when they are colonized by embryonated egg. That is sometimes due to contamination of the food, even we might get infected with this cysticercus. If the portion of the pork is eaten by the man, the bladder is dissolved by the gastric juices inside the man, and this gastric juices causes the albinous material to swell up, and exvagination occurs, and from where the worm survives. And once infected, the worms gain sexual maturity within 2 to 3 months, and it survives for almost 25 years in the vertebrates. It can be humans or it can be any vertebrate it is infecting. So this is how the life cycle looks like. You can just go through that. And the effects of parasite, discomfort, pain, indigestion, vomiting, constipation, loss of appetite, dysentery and diarrhea, nervous disorders like nervousness, insomnia, you don't feel sleep at all, insomnia, nausea, and epileptic fits, that is fissures or fits. Some people have fits is because of this parasite tinea. Though tinea is comparatively less dangerous than cysticercosis caused by infection of bladder worm. And so there are different cysticercosis. Again, when it reaches the vital organs like liver, eyes, and the brain, they cause fatal diseases because they are going to cause blindness, and the brain develops epilepsy. Epilepsy is nothing but again fits, fissures, or the seizures that you call them. So this is all about the platyhelminthes. Any doubts, Ma? We have completed 3 classes, Turbularia, Trematodes, and Cestodes, and we spoke a little bit about the different parasites of Trematodes and Cestodes, and we also talked about the clinical importance of, not clinical importance, scientific importance of planarians, and how uncooked food can cause different teniasis and different diseases that occurs in the humans, and of course the cattle also. So with that we complete platyhelminthes, and tomorrow we'll do the next topic. Yes. Okay, so that's about the platyhelminthes for today's class, and hope you have understood. Any doubts?
No, ma'am.
So shall we close the class today? And
Thank you, ma'am.
welcome. So meet you all tomorrow at 4:30 again, right? See you all.
Okay, ma'am.
Okay.
Ma'am,
Yes,
Ma'am,
yeah, tell me.
ma'am, give me some important questions for examinations, ma'am.
Yeah, important questions as such, because I'm not going to prepare the questions, so I will not be knowing exactly what type of questions that examiner is going to give. Because as and when I'm teaching y'all, I keep telling y'all this is some importance, that is some importance. So you just make a note in your rough book. So like, for example, In Platyhelminthes, they will ask you all about Taenia solium, one, characters of the Taenia solium, how the external morphology looks like, or they will also ask you to talk a little bit about protonephridia for short questions. Protonephridia, you can just make a note of it. They will ask you to talk about protonephridia. They will also talk about the larval forms of the trematodes and the cestodes. We will have to talk about the trematodes and the cestodes. A little bit about fasciola hepatica and life cycle. Both the life cycles are important in cestodes and trematodes. So yeah, those are some important questions that you can talk about. Okay.
Okay,
Ma'am,
ma'am.
what are previous topics, ma'am?
Previous topics,
Means important questions, ma'am.
I had already discussed about that when I was talking, taking my class, Noma. So each time I can't keep telling the same topics, right? Because time is important. I have to leave. I have some important work. Okay. Meet you all tomorrow. See you all.
Okay, ma'am. Thank you
Yeah.
so much.
Thank you.
Ma'am, thank you.
Welcome. Right.